Welcome everyone. This is Mani Attirajan. I am a DMF reviewer from the Division of Lifecycle API. In the next 21 minutes or so, I will be describing the FDA's expectations in the peptide DMF with respect to peptide API sameness and related impurities. Therefore, my poster title is Synthetic Peptide APIs of Generic Complex Drug Products. Our recommendations for API sameness and related impurities. In the beginning, I will go through a few introductory slides for the benefit of the new applicants. Later, I will focus on the main topic of our recommendations for API sameness and related impurities. In this slide, I'll give you what are the key criteria required for a generic drug. For an example, if you're applying an ANDA under Section 505J, you must demonstrate that your proposed generic drug should be equivalent to RLD. For example, it should be pharmaceutically equivalent, bioequivalence, and your product should be adequately labeled. So what are the pharmaceutically equivalent means? It means your drug substance should be as same as the RLD. You should use the same dosage form. The route of administration is same as the RLD. In general, it meets appropriate standards of strength, quality, purity, and identity. As a DMF applicant, we will only worry about the drug substance sameness. The rest of the things are related to drug products. So these are the three criteria if you're applying for a peptide drug product. So before we go into the details, what are peptides? As per FDA, it considers of a polymer of 40 or fewer amino acids. And the FDA believes at this time that only synthetic peptides are eligible to be approved as the generic under Section 505J of the FD&C Act. So if you talk about the DMF, as I told you, we only worry about the drug substance sameness. So I'm going to go show a couple of examples of peptide drug before I go into API sameness. Here are a few examples of generic peptides: bivalirudin, calcitonin, teriparatide. Bivalirudin consists of 20 amino acids. Calcitonin, it's a cyclic peptide, consists of 32 amino acids. And teriparatide consists of 34 amino acids. So we have seen a few examples here. Is there any regulatory guidance available for peptides? In general, there is no peptide guidance. There is no ICS guidelines for peptides. However, we have a draft guidance for industry, especially and as for certain highly purified synthetic peptide drug products that derive from the rDNA. This guidance applicable only to liraglutide, glucagon, nesiritide, teriparatide, and teriglutide. We also have a product-specific guidance as for glatiramer acetate, semaglutide, and linaglutide. In the case of semaglutide and linaglutide, it is similar to our draft guidance for the synthetic peptide drug products that refer to listed drugs of rDNA origin. Now we have a draft guidance for synthetic peptides for RLD of rDNA origin. We can utilize this guidance to establish the API sameness of the peptide as well as related impurities present in it. So let me discuss the gist of this guidance with respect to API sameness and the impurities. In general, this guidance recommends how to establish API sameness as well as how to qualify your related impurities
by comparing the RLD product. Under the active ingredient sameness, the guidance recommends that you should provide a complete characterization of your drug substance or drug product. That means the primary sequence of your drug substance or drug product, as well as the physical chemical properties of your drug substance or drug products. If necessary, you can compare it with the RLD sample. That would be very great. The guidance also recommends you to submit secondary or higher order structures of the drug product. Similarly, if the drug product exhibits any oligomer or aggregation states, you should provide it. Or you can also compare these studies with the RLD. In some cases, you may need to submit a biological activities, your drug product. It depends on the case-by-case scenario. Please remember, in the DMF, we require only the primary sequence and physical chemical properties of the drug substance. The higher order structures, oligomer states or aggregation states, biological activities are only required at the ANDA applicant level, not at the drug substance or DMF level. Please remember that. In the case of related impurities, the guidance recommends that the applicant to do the impurity profile comparability studies of their drug substance or drug product against the RLD. Based on these studies, you can propose the limits for the specified impurities. When you're planning to do these studies, please consider the criteria which I'm going to present here. Suppose if you're getting impurities that are common between your drug substance and the RLD, the acceptance criteria should be not more than those observed in the RLD. So these studies, we always recommend you to do the minimum of 3 batches of generic drug substance and minimum of 3 batches of RLD. Suppose if you get any new impurities in these studies in your drug substance, which is not observed in the RLD, then the limit should not exceed 0.50%. Further, each of these new impurities present at the level of greater than 0.10% should be identified and justified. The guidance also recommends you to utilize sensitive and high-resolution analytical methods, example, UPHPLC, HRMS, etc., to detect and characterize peptide-related impurities in your proposed generic peptide in comparison to RLD. Please identify and report each specified and unspecified peptide-related impurities that are greater than 0.10% of the drug substance. If that is the case, your analytical method LOQ should be less than 0.10%. So as you can see, the guidance expects you to submit a physical chemical properties of the drug substance. In the DMF, what are the things we require under the physical chemical properties of the API? Of course, we need a description of the drug substance, solubility of the drug substance under the different solvents and different pHs, and hygroscopicity of that drug substance, and potential isomerism if anything exists, or chirality of that API. If that is the case, you should provide a minimum specific optical rotation of that drug substance. Of course, polymorphism. And I remember most of the peptides are amorphous, so you may not need to submit it, but that is not the case for always. You need to submit a melting point Well, in some cases, peptides are amorphous, so you may not need to submit those things in that circumstances. In some cases, you may submit different studies, which depends on the nature of the API. In terms of characterization, again, with respect to DMF, we will need a complete characterization of a peptide sequence. For example, basic characterizations like IR, UV, mass, and NMR spectroscopy. It's better to provide a proton and C13 NMR together. If needed, you can also provide the 2D NMR studies and also amino acid analysis. Since our peptide target peptide drug is a chiral amino acid, contains chiral amino acid, so we require definitely a chiral amino acid analysis. Definitely we need amino acid sequencing. You can use Edman degradation and or MS-MS sequencing. Some cases we may need additional studies. It depends on the API. In order to identify the potential impurities in your drug substance, you need to understand the origin of the impurities. Remember, differences in manufacturing process may result in differences in the related impurities. For example, chemical synthesis. If the DMF manufacturing process uses chemical synthesis, you may expect impurities from starting materials like amino acids, reagents used in the process, catalysts, solvents used in the process, intermediates arises from the process, and also byproducts from the process. In addition to all the above impurities, you also expect degradation products. In the case of recombinant synthesis, you may expect fermentation and cell culture media components, and sometimes you may expect residual DNA and cellular proteins used in the process. You may also expect bacteria, fungi, mycoplasma, viruses may, as side products or impurities are derived from these, fungi and bacteria, viruses. And also you can expect column materials, impurities from column materials, and of course the other degradation products. In the recent days, especially in the DMF, most of the APIs are manufactured using a solid phase peptide synthesis. So let's see what kind of potential impurities arises due to solid phase peptide synthesis. So impurities may result from the insertion, deletion, and modification of amino acid sequences or residues. So these are the impurities you can get it during the solid phase peptide synthesis. Those impurities can be a process or degradative in origin or both. I'm going to give you a few examples, key examples of related impurities one can expect during the solid phase peptide synthesis. You can expect a proteolysis type impurities, deamidation impurities, oxidation impurities, reduction impurities. Sometimes you may get resinization leads to impurities, deletion impurities, truncation impurities, or addition of amino acids in the chain. Sometimes you may get incomplete deprotection impurities, or if the peptide consists of a disulfide bond, and you may get a disulfide exchange impurities. So these are the major impurities, and you may also get different kind of impurities based on the method or controls you are using in the process. You may also get impurities due to our manufacturing process and the storage. Now you know what kind of impurities one can expect based on the manufacturing process. Now you can establish your impurity profile of your drug substance. And also you can compare that with the RLD using the same analytical method. When you do the impurity comparability studies, please consider the following. Once you've done the studies, please demonstrate that the impurities common to both the proposed drug substance and the RLD are present in the proposed drug substance at the same or lower levels than in the RLD. Analyze and characterize any new impurities in the proposed drug that are not common to the RLD. Please be aware that the impurity profile of your drug substance is not always same as that of RLD. It depends on the process involved and controls involved in both the process. As I insisted earlier, please conduct each study on a statistically meaningful number of batches. It is also recommended that the proposed drug substance be tested on or near release at the end of the proposed shelf life. Similarly, the RLD batches of different ages to be tested prior to expiry. This will give you a complete picture of your impurity profile and also the RLD impurity profile. Please provide sample ages and the dates of the studies. For these studies, we would always recommend you to use a multiple orthogonal validated methods. That is very useful to determine the structure of at least gives the idea of the structure of the impurity and purity of the impurity you are dealing with. In the case of orthogonal analytical methods, especially in the DMF, we recommend you the following. If you have a related substance due to deletion, insertion, truncation, proteolysis, substitution, functional group modification, or disulfide modification, deamidation, acetylation of amino functions. So all these related impurities due to these modifications, it's better to use LCHRMS, MS methods. Those will give you the clear idea of what kind of impurities we're dealing with, and also gives you the structures of the impurities. Based on the key points I mentioned with respect to related impurities, here I present a simplified scenario of how to establish your drug substance impurities and their limits based on the impurity compatibility studies. Here I presented the list of generic drug substance impurities as well as the RLD impurities. In this case, impurities A, B, C, D are common impurities. Impurity E is a new impurity which is not observed in the RLD impurities. However, the limit proposed for the impurity A and impurity E is not acceptable. Here, the impurity A is observed at 0.50% at the RLD level, but the proposed limit is more than the 0.50%, so that's why it is not acceptable. You should always propose same or less amount you observed in the RLD. In the case of impurity A, which is a new impurity, but you should propose a limit only at the level of 0.50%, not more than that. In the case of AD unspecified impurity, the proposed limit in the generic substance is not acceptable, even though it is same as the RLD level. The reason is in the RLD level impurities, it went through clinical studies and it is qualified through the clinical studies. In your case, it is not because of the immunogenicity risk. We don't allow not more than 0.10%. So please remember, when you establish any new impurity, always you should be equal or less than 0.50%. Any common impurities should be less than or equal to RLD impurity level. Any unspecified impurity, we always recommend you to set the limit at equal or less than 0.10%. Please make a note that we extend these recommendations to all the peptide DMF API DMFs, whether the corresponding RLD is derived from the chemical synthesis or RNA derived. Please also make a note that any additional data can be requested for the impurity E from the ANDA applicant referencing this DMF. So please be aware of that. So takeaway points from my presentation is please remember API sameness as well as impurity profile compatibility studies on the proposed peptide drug substance are very, very important part in your drug development. Also remember each peptide API has its own challenges. Therefore, DMF applicants need to evaluate individual situations and apply these recommendations accordingly. Finally, thank you for your attention. If you have any questions, please send your questions regarding this poster to DMF Workshop 2021 at fda@hs.gov by February 15th, 2021 for inclusion in the poster question and answer session on March 3rd, 2021. There are a follow-up webinar for both posters presentations on April 9, 2021. You can also send questions to the above email by 3/19/2021 for the webinar. Please also refer to the following presentations on March 3rd and 4th for additional information. The first presentation is regulatory considerations and demonstrating complex API sameness. The second one is safety evaluation of drug substances, impurities in generic. Both are related to this presentation, so please refer. Thanks again for your attention. Thank you.